right, nice to see you. I appreciate you being here when it's sunny and lovely outside and not sweltering hot. Who has a joke? Hot or not? Don't worry, I'm not going to make latecomers tell the joke. Although it could be a good policy. Come on, someone's got one. You heard what happened when I tried to tell one last time. You really don't want to experience that again. Hello, anyone? Ah, excellent. Okay. You guys arranged this ahead of time, right? That it would be someone, wherever I went, someone on the other side of the room would raise their hand. More steps. Okay, hold on. Nice and loud.
What's Peter Pan's favorite place to eat out?
What is, and I suspect the word order is important. What's Peter Pan's favorite place to eat out?
Wendy's.
I like Tones, Tinkerbell. Yeah, okay. Is nothing sacred in childhood? Is nothing safe from lewd jokes? What's the answer? No, nothing's safe. So last time, we're still talking about male sexual anatomy. I think we're going to finish today. And last time I was reminding you that the neuroscience is such that in the brainstem, there are serotonergic neurons that send their axons down the spinal cord, and they originate in the paragigantocellular nucleus of the pons and medulla. And that those serotonergic fibers, they go to every spinal cord level. It's a pervasive influence. But the thing we're interested in is that once it gets to lumbar and sacral regions, it inhibits the reflexes that drive ejaculation. And so, and erection. And so it sometimes happens that someone's taking SSRIs and it impairs their ability to get an erection. And I told you last time, if you try different SSRIs, one that has that effect in one person might not have an effect in the other. So it's a matter of trial and error. The other thing I pointed out to you is that that because SSRIs take weeks to have any effect on depression, that it's got to be somehow the nervous system is responding to or compensating for those weeks of serotonergic synapse enhancement. One evidence of that is because the serotonergic fibers inhibit erection ejaculation, it sometimes is helpful for some men who ejaculate too soon, before they want to, before their partner might be ready for them to. Sometimes taking SSRI helps them to last a little longer before they ejaculate. And the proof of how it works is that in that case, you don't have to take it for weeks. Just taking the drug half an hour before sexual encounter will be enough to get the complete effect, which shows you that it works right away, right? Soon after you put it in the nervous system. So if that was the only way depression was lifted, it should work instantly. And as you may know, there's a big effort trying to find antidepressant drugs that work right away, not having to take weeks, because of course, depression can be dangerous because there's always a risk of suicide for people who are depressed. And so, and this will probably be the only place where I get to say that there is a pernicious myth that if people want to commit suicide and you try to stop them, they'll just go ahead and do it anyway. And it's been proven that's totally untrue. Most people who make a serious attempt at suicide never try it again. And if you want something really harrowing, people sometimes would jump off the Golden Gate Bridge as a way of killing themselves. It almost always worked. But there are a few people who survived it. And every one of them said, as soon as they jumped, they wished they had not. So those thousands who jumped and died, what were their last thoughts? Oh no. So, number one, intervening when someone is suicidal is effective. It's important. It's your job as a person. Okay? Don't shrug it off. Okay. Now, these same mechanisms, let's compare what we've been talking about in males to the female anatomy. Again, this mid-sagittal view at the midline. And I'll just remind you that of the anatomy, here's the bladder, here's the urethra, here's the vagina, anus. The cervix, of course, is the beginning of the uterus, which is up there. And the point we're making here is the same the same autonomic stimulation that prepares the male genitalia for sex behavior, which is which? Prepares the male genitalia for sexual encounters, is a parasympathetic point. It's also the parasympathetic nervous system that has to be working to prepare the female genital system for intercourse, to facilitate sexual lubrication from the walls of the vagina and vasocongestion and things like that. And just as sympathetic stimulation, think of it as stress, can inhibit erection in men, sympathetic stimulation, again, stress, inhibits this process. In women, it may make the sexual encounter less pleasant, right? And so when we talk about sexual disorders, almost all of the sexual disorders that people commonly complain about, they're almost all due to stress. And from a neuroscience perspective, one aspect of that is activating the sympathetic nervous system is not good for getting the genital system ready for sex. And what's the other thing I have? Oh, sorry, should have shown that one first. And the same nitric oxide is a transmitter of both. I mentioned to you that one way of telling whether erectile machinery in the penis is still working is to ask whether men who, most people, especially if they're not setting alarm, or even if they're setting alarm, if it's set for the same time every day, the last sleep episode of the evening tends to be a REM sleep episode. This is why when you wake up, especially if it wasn't with an alarm, you might remember a dream for a while because you just came out of a REM episode. And for males, if they did have a REM episode, they probably still have an erection from that. Yeah.
But for the in males, it's parasympathetic until the end,
Mm-hmm.
and then when ejaculation happens, it's a sympathetic reflex.
Correct.
You said that females are similar in the parasympathetic way.
Mhm.
Do they have the sympathetic reflex? Do that yet?
We're going to talk about that in a moment. We won't see it as a sympathetic arousal, although it happens, but we'll see one consequence that is driven in both sexes and it's the same response in both sexes. Good, yeah.
Type functions in women.
Yes, absolutely. So in both sexes, right, it is having that inhibitory effect. And so, yes, it can also inhibit, and where it would be seen most is that there'd be less erection of the clitoris. And again, some SSRIs do for a particular person and some don't. If someone found an SSRI that never caused that side effect in anybody, that'd be the only one out there. But it's more of a random decision, apparently. Other questions? So at ejaculation, so ejaculation referring to this rhythmic peristalsis and expulsion of semen from the male genital system. When that happens, the sympathetic nervous system becomes suddenly active, causes stimulation of the vas deferens. So there's this peristalsis that is shoving sperm, pretty much purified, but so far incapacitated sperm, into the base of the penis. The autonomic nervous system is also squeezing semen out of the seminal vesicles and causing the prostate to add fluid. The semen from the seminal vesicles is viscous and white, and the material from the prostate is more clear and slippery. But as we talked about last time, the components have to come together to capacitate the sperm so they have become active and will have some way of working. Another thing that happens at the same time as that is those striated muscles at the base of the penis, the ones that squeeze out the last bits of urine when you're urinating, those also become active and they contract rhythmically. And so this will cause the semen to come out in rhythmic spurts. We'll show you a picture of how the timing's like in a moment. And among the muscles that are used for that are the bulbar cavernosus and the ischiocavernosus. Bulbar cavernosus, also sometimes known as a bulbar spongiosus. So we talked about that and showed that earlier. Sometimes the external anal sphincter will also contract rhythmically during that period. And so for some men that might happen, might not. For some men, having something, a finger, a dildo, something like that, in the anus at that time can accentuate that response and bring some men enjoy that more. Questions about that? And these same muscles, that's the next slide, are also rhythmically active in females. In many females at orgasm, there'll be this rhythmic contraction of the external muscles. And don't, hold on. Oh, I see. I thought I had it. Yeah. No? Okay. We'll show you that in a moment. And so many women also experience the rhythmic contractions, which they might experience as a rhythmic constriction of the opening of the vagina, and or they might experience as rhythmic contractions of the anus. And so measuring these muscles in either sex is often a pretty reliable indicator of when an orgasm has happened. I've told you before the muscles are weird in that we do have voluntary control of them over them, but there are times like this when the spinal cord circuits override the voluntary control and cause this rhythmic contraction to happen without your conscious effort at all. On the other hand, it's not entirely push-pull. So there are some communities where birth control is forbidden, so certain Amish communities, for example, and yet if men don't want their wives to get pregnant, apparently in those communities, there's a practice of exercising bulbar cavernosus muscles, making sure with Kegel exercises, making sure they're nice and strong. And then at the point of ejaculation, the men will purposely bear down on the bulbar cavernosus muscle and prevent the ejection of semen. And so where does it go? Well, it's got nowhere to go except back up that same duct and into the bladder. And apparently it's not particularly pleasant sensation, but on the other hand, they still get to have sex and not, and reduce the chance of their partner getting pregnant. Reduce, but not, but not foolproof. And then afterwards in a urine flow, the urine will be milky because the semen is still in there and they're getting rid of it. So whether you do that or not, whether you want to use that as birth control, and I don't recommend it, or not, it is a good idea to keep the muscles in good tone, and so you can exercise them in either sex. For most women, when the rhythmic contractions of the constrictor vestibuli, that's their version of bulbar cavernosus, bulbar spongiosus, when that happens, for most women, that's pleasurable, and they enjoy feeling that while there's a penis or dildo in the vagina. Yeah, normally, at ejaculation, the urethral sphincter closes the bladder, so there's nowhere to go but there. But as I said, if one contracts these muscles surrounding the base of the penis hard enough and long enough, it will, you'll have also known as retrograde ejaculation, where it goes back up to the bladder. These striated muscles are controlled by the spinal cord. And there's a particular nucleus in the sacral spinal cord in humans where these motor neurons reside. And send their axons out to control the muscles. And they're in a nucleus known as Onuf's nucleus, named after the guy who first described it in 1899, and then another paper in 1900. And the reason it came to his attention was because he was looking at polio victims. And when he did autopsies of polio victims, he noticed something interesting, which was that in those victims of polio or Lou Gehrig's disease, either one, in those victims who were able to remain continent and control their bladder till the very end, Onuf's nucleus was always intact. But in those individuals who became incontinent, incapable of controlling the urination before they died, those motor neurons were often dead. And so he reasoned, he didn't have the anatomical tools to show it yet, but later people would. He reasoned that these motor neurons must be controlling the striated muscles of the pelvic floor and of the penis. Questions about that? And what's really interesting, is in Lou Gehrig's disease, also known as amyotrophic lateral sclerosis, that is typified by lots of motor neurons dying throughout the spinal cord. Onuf's motor neurons never die. Something about them is special that they are not susceptible to Lou Gehrig's disease. I've looked at spinal cord sections where it's almost impossible to find any motor neurons, and then I get to Onuf's nucleus, and they're all there, looking bright and happy and healthy. And so that's one reason why people with Lou Gehrig's disease typically remain continent. Okay, enough neuroscience outside sex for there. Questions about any of that? And here I'm showing you the pattern. Here this is electromyograms where they put tiny little electrodes, too small to be uncomfortable, into the bulbar cavernosus muscle, and then recorded activity as the person probably masturbated and ejaculated. And they could note that the spurts of ejaculation happened about here. And you can see, number one, the contractions perfectly coincide with the ejaculation, but they also continue for a little bit after there's no more ejaculate coming out, right? So, bless you, twice. And so it's intended to make sure that the whole contents of the base of the penis, the little mixing chamber at the base of the penis, that it all gets pushed out. The force of the ejaculation, it might have been impressive for Portnoy in the novel, but in fact, it's not very important. It just has to get out because after that, for the sperm to get upstream, they're going to have to be motile and they're going to have to be working their way up to the cervix, through the cervix, and into the uterus. And if a pregnancy is going to ensue, then they have to fertilize the egg where? Pardon? In the oviducts. If the egg's already down in the uterus and the sperm get there, no pregnancy is going to happen. An embryo will be formed and it will be dividing, but the walls of the uterus will be shed before it can implant. And related to this business of the rhythmic contractions of the striated muscles of the pelvic floor in women at orgasm, number one, not all women who have orgasm experience that. So it's not a certainty. So no, you cannot be sure whether your partner had an orgasm based on that. Number two, in some women, the rhythmic contractions are accompanied by rhythmic expulsion of fluid. And I don't know, in the 90s or so, I suppose, there was this big controversy. Do women ejaculate? Well, first of all, some women absolutely expel a lot of fluid in a rhythmic fashion when they're having orgasm. That's, empirical fact. Whether or not you call it ejaculation or whether or not you call it an ejaculate is really just a matter of semantics, but it certainly happens. And people did experiments like this to show that most of that fluid that comes out is indeed urine. And so hair splitters might say, well, since there's no semen contribution, no prostate contribution, it's not really ejaculation. Well, says who, right? So a man who has an ejaculate and there's no sperm there, is it still an ejaculate? Right. Still ejaculation? Sure. that rhythmic, if you consider the ejaculation to be that rhythmic aspect of it, it's there in both cases.
Out unconsciously or consciously?
Let's see. So there are plenty of women in which it happens and they have no conscious control over it. And for some women, they're embarrassed by it, especially if they didn't know it was going to happen. So another thing that's good advice, whatever sexual problems you have, the best therapy is to masturbate. We'll talk about this more. So, practice when you're alone. And so for some women, when they have an orgasm and ejaculate, they might be embarrassed by it. And because it's not typical, but there's nothing to be embarrassed about. I'll remind you that urine is sterile, right? Unless the person has a bladder infection, there's no health risk involved. Yeah, okay. Get on the sheets, so do menses flow, but all that stuff can be dealt with. And so if my advice would be for women who do experience that, some women try to clamp down at that time to try to prevent the flow. And for some women that works and some it doesn't. But the main thing is communicate, right? To make sure your partner knows it's going to happen and don't worry that they've done something wrong or that you're hurt or something like that. Yeah. Well, there's probably some contribution to here they're suggesting this periurethral gland that is near the mouth of the vagina. And so it's a gland. It's still, it's there. You can see it. It's pretty small. So it may very well be contributing, just like semen in men is a mixture of fluids. This too is a mixture of fluids. But the volume is such that it can't be responsible for most of the volume. Just not big enough. Yeah, Ethan. Mhm. One and the same. So the constrictor vestibuli is a striated muscle of the pelvic floor, which in males is called the bulbospongiosis or bulbocavernosus. Yeah. Pardon?
Sympathetic nervous system, is it in control of this?
I'm not going to say that it's in control of this, but it is activated at this point in both sexes. And oh, I see. Yes, it's sympathetic activity that is causing that rhythmic contraction to happen. Yes, it's being driven by them. And I think we already talked about this as a way of keeping track of female sexual arousal. And the main take-home message is during sexual arousal, because of the parasympathetic activity in women, the walls of the vagina will become fuller of blood, and there'll also be this pulsing that's going on. And so people have been able to correlate the filling of the walls of the vagina and that pulsing to sexual arousal. And I'm going to skip this because we'll talk about it later. We will get back there. The last thing is something to talk about that's pretty much regarded as a phenomenon in males. There's a so-called Coolidge effect. So this is based on a joke that was probably never very funny. Cal Coolidge was a president. You probably knew that, Calvin Coolidge. And apparently he was a very stoic man, hardly ever showed any emotion. Groucho Marx had a joke that when Cal Coolidge died, they came and told his wife, Mrs. Coolidge, the president, has died. And he said that she asked, "How can you tell?" And there was a joke going around that you've heard in one form or another, which is the president and his wife are touring a poultry farm, and he's going first, and he goes someplace, and they show him all the animals in the yard, and he says, "So I see lots of hens, but I only see one rooster here. Do you mean to tell me this one rooster services all of those hens?" And oh, pardon me, it's Mrs. Coolidge. Yeah, I can't do this. And she says, "You mean that one rooster services all these hens?" And they say, "Yes, First Lady, whatever, Mrs. Coolidge, that's the case." She says, "Be sure to point that out to the president when he comes by." So then later they show him the president, the same thing. And they said, "And your wife wanted us to point out that one rooster services all these hens pretty much every day. And he said, well, excuse me, is he servicing a different hen each time? They said, yeah. Well, point that out to the first lady. I told you it was a weak joke. Well, but the joke got compounded because back in the late 1950s, Frank Beach, we'll run into him again later, and a graduate student had been studying this phenomenon in rats, which is this, that you can take a rat and give him a female partner, and he will copulate and ejaculate 3 or 4 times in the course of an hour or so, and then he'll show no interest in her. And you would think that means that he's sexually sated, that he doesn't want any more sex. However, if you now take out that female and drop a new female, and he immediately starts copulating and will ejaculate with her several more times. And in fact, you can do this several times. And so what we're seeing here, I can't remember if this is in rams or rats, where they present the same female repeatedly to the male, and he ejaculates right away the first time he sees her, and the second time, the third time less so, fourth even less, fifth, it takes him 17, 18 minutes before he actually ejaculates. But the same male, if you present a different female each time, you can see there's virtually no change in how quickly he mounts or how quickly he ejaculates. So the Coolidge effect is where an animal, typically a male, that seems to be sexually sated with one partner will recover his libido when presented with a new partner. Everyone gets the idea? So they'd been studying this in rats, and they were going to do a presentation at the American Psychological Association, and they decided, because that joke was going around, they decided, we're just going to call it the Coolidge effect, and we'll get a laugh from the audience. So they present the data, call it the Coolidge effect, use the term several times, each time expecting people to start, and nobody did. And so now it's embedded in the literature. Questions about that? And what I like to say, I had a reporter once ask me, is there a Coolidge effect? Does it work in humans, or is it only in rats and goats and things like that? Oh, I said, of course it is, because you won't understand this way. If it didn't, nobody would ever buy more than one copy of Playboy. And yet they sell millions of them every month. Right? Yeah. Well, I thought it was funny at the time. She used the quote. Or he used the quote, I don't remember. Questions about any of that? And that you can read on your own time about this guinea pig who happened to find himself with lots of females and just exhausted himself, poor dear. Yeah. It's a great question. So the evolutionarily thinking folks that think that males, that evolution has primed them to spread as many gametes to as many people as they can. The idea being, if it's a fresh uterus, then you've got a fresh chance of having a pregnancy result, right? They would say, well, women don't work that way. And in general, I would say that most women do not, right after having a sexual encounter, right away want to have an encounter with someone new. But of course, some women do. And I don't think there are any women who would say that having been with one partner for a long time, they wouldn't find the idea of being with another partner exciting, right? Even though our culture tells them they shouldn't, right? They should be totally faithful and only be interested in one person. Yeah. Maya.
It was a cam girl or something one weekend, but there's a documentary where she endeavored to sleep with 1,000 men in one day.
Yes. Yeah.
She did do this, and actually there was a bunch of controversy because a lot of the men in it were cheating on their partners when they went to go do that, and then it got put on a documentary.
Yeah. So, and I mean, there's a famous story of the Emperor Claudius's wife supposedly, having a lot of sex with a lot of men in one day. In this particular case, the porn star was doing it to get attention, and as long as everyone's got a condom and everyone's consenting adults, okay. I bet she had loads of lube around. And I doubt that, I mean, could she really wait for each of the men to ejaculate? I mean, if so, hurry it up. We got to go. We got a record to break. And I can't remember, she definitely got close to 1,000, but yeah. So yeah, it could be done. I don't think she was doing that because she wanted lots of partners. I think she was doing it because she was gonna make money at it. Right? I mean, now that we are in the attention economy, where you can make a fortune if you can get enough people's attention, whether you're talented or not, whether you do anything interesting or not, right? There's money to be had there. Okay, well, let's have a coup. We're having talked about the sexual anatomy of females and the sexual anatomy of males, by which you notice things are not the same. And now we're going to talk about how those differences arise. And that's the process of sexual differentiation. So, as we'll see early in fetal development, the urogenital system looks the same in whatever sex, in both sexes. But what's going to happen is in the course of fetal development, starting in the first trimester, actually, in the course of fetal development, some fetuses are going to take one directional path and some another. And so the two sexes start becoming different, one from the other, and they become increasingly different as gestation goes on. That process of coming to be different from the initial state, being male-like or female-like, is sexual differentiation. Good. Absolutely, yeah. I mean, yeah, I mean, you're going to be sick and tired of talking about intersex phenotypes. I mean, maybe I overdo it. What we're going to see is that genes play an important role in sexual differentiation to take place, chromosomes and genes, and that hormones are absolutely crucial. So this will be our time to talk a little bit about the basics of hormone action as we'll get there. And so that's the stuff we'll talk about. The first question, you already know that in many mammals, certainly in our species, sexual differentiation is dependent on sex chromosomes. So we can talk about, as you probably know, if the embryo is fertilized by a sperm carrying a Y chromosome, it normally will become a male phenotype. And if it's fertilized by a sperm carrying an X chromosome, it'll normally take a female phenotype. And so that's why females have two X chromosomes and males have one X chromosome and one Y. That's pretty much common knowledge. You probably know that already. And because females have only X chromosomes, every egg carries an X, right? They don't have any Ys to contribute. This means that it's the male that will determine whether the offspring will go down a male pathway or female pathway. And the beginning of that determination is based on the sex chromosome. And every male has both an X and a Y, and therefore some of his gametes carry his X and some carry his Y, and the ones that have his Y are going to result in a male. So when this happens, we'll talk about this in more detail later, as the sperm is going upstream, it is changing its composition and eventually reaches the outer layers of the egg, because it has this gelatinous outer layer, and it has to find its way through there, and then it has to be admitted by the ovum. And the ovum will only admit one sperm, and it is going to make a decision about which sperm to admit. Oh, and it's the only time I've actually said the funny thing right when the opportunity came. We'd taken our daughter to see cats, which was a terrible mistake. She loved it, and so we were stuck with it for years. She was like 4 or 5, and we got there ridiculously early because it's in the Bay Area, and you never know how long it'll take you to get over the Bay Bridge. So we're there ridiculously early. The theater's practically empty. There's only, but there's a couple sitting in front of us, and Tessa says, Dad, is it true? Did you decide whether it'd be a boy or a girl? And I said, yes, Tessa, I did. And she said, how did you decide? And I said, well, Tessa, I thought about it long and hard. And what I did, the lady sitting in front of us bent over laughing. So it was like, oh, yeah, how great. I thought of, I mean, it came to me at the right time. Anyway, so to speak. So what will happen then is the fertilized egg, also known as a zygote, or we can call it a conceptus, will start dividing. So we're starting to make an embryo. And first it makes more or less homogeneous massive cells. If you want to know more about that process and what happens thereafter, you've got to take Neuroscience 416 next fall. And eventually that sphere of cells will form a hollow place inside that's filled with fluid. And we call that the blastocele, the insides. And this entire thing is a blastocyst, so it's an early embryo. And in mammals, unlike other vertebrates, in mammals, once the blastocele forms, there's also this clump of cells within it. And the entire body is going to arise from this clump of cells known as the inner cell mass. That's how it's done in mammals. And the rest of the embryo, the blastocele and that membrane, is going to form the placenta to maintain the embryo in the uterus through the pregnancy. Well, in terms of reproduction, another thing I want you to know is gametes are strange cells. They're not just strange in that they can make a new person, not just strange in that they're haploid instead of diploid, but their origins are strange too. So in the embryo, in the developing embryo, there are things, these cells that arise that are known as primordial germ cells, not primordial, primordial germ cells. And they look like any other cells, but in fact, they have distinctive characteristics, and these are going to be the source of gametes. And after the embryo has formed the beginnings of gonads, then these cells in circulation, they're going through circulation constantly, but when they get to the gonads, some of them will embed there, and they're the ones who are going to divide to form gametes. So think about this for a moment. You began life as a primordial germ cell in circulation in your dad as an embryo, and another part in your mom as an embryo. And the whole point of natural selection and evolution is those two primordial germ cells eventually became gametes fused. And if they're going to reproduce, they've got to produce you. And the first thing you'll do in order to give them grandchildren is you've got to make primordial germ cells. So that in real sense, the whole reason of having a body is germ cells making a machine to propagate more germ cells. And you're just a device for getting germ cells to go from one generation to another. I hope you don't feel bad about yourself, but this is how it is. Okay, enough philosophy. So, where do the gonads come from? Early in embryonic development, all the sexes look the same. There are these clumps of cells that are near the kidneys, the developing kidneys, and we can refer to them as indifferent gonads because in males they're going to make testes, in females they're going to make ovaries, but at this phase they don't look like either. And so we call them indifferent not because we don't care about them or they don't have any value, but just because we can't tell them apart. And attached to these indifferent gonads are two different systems of cells that connect the gonad to the midline and then to the outer layer of the body. And in fact, these two sets of ductwork, eventually they'll form things that are hollow. So that's why we're calling them ducts. Eventually, when they reach the outside of the body. They come to a point on the ventral surface between the hind legs. And eventually the tissue there will make a little bit of a nubbin. We can call that a genital tubercle. It's just a little bump. Doesn't look like a clitoris, doesn't look like a penis. It's something in between. And at this phase, males and females look exactly alike. But they're not. Because and these ductworks are named by the people who first described them. They're the Mullerian ducts and the Wolffian ducts, and so they're there in parallel. And the Mullerian ducts are going to give rise to the female reproductive system, and you should be glad that Mueller was the one who discovered it, because then you can, as a mnemonic, remember that M starts with M, Mullerian and mother. So you can't be a mother without Mullerian ducts. And the other set of ducts described by Dr. Wolf are known as Wolffian ducts. And if you think of wolves, you know, the caricature in cartoons of males being wolfish and wolf whistles and things like that, that'll make it easy if you remember. That's going to make the male reproductive tract. And what happens next then is if a Y chromosome is present, then the indifferent gonads start developing. I guess I told you all that. If a Y chromosome is present, then the indifferent gonad starts developing as a testis. And it does this because in the indifferent gonad, there's both the outer layer of cells, the cortex, and the inner layer, the medulla. And if a Y chromosome is present, it'll be the cells in the cortex, in the medulla, that will divide and make testes. And in the absence of a Y chromosome, it'll be the cells in the cortex that divide and form ovaries. So once that starts happening, once the indifferent gonad starts looking a little bit like a testis or a little bit like an ovary, then those duct systems that are going alongside start changing too. So when a Y chromosome is present, the testes develop and the Mullerian ducts fade away. And the Wolffian ducts persist and are going to provide a pathway for gametes from the gonad to reach the outside world. And I'll give you the names of those structures in a moment. In the absence of a Y chromosome, the Wolffian ducts wither away and the Mullerian ducts develop, and they're going to provide a pathway for gametes to get to the outside world in females. And this is all happening in the first trimester, right? So the fetus is teeny tiny, half an inch in humans at most, and yet already these decisions are getting made. And the crucial gene for having gonads develop as a testis were identified in the late 20th century. And the gene is known as the sex-determining region of the Y, or SRY. So females normally have 2 X chromosomes, and X chromosomes do not have an SRY gene. Males, on the other hand, have 1 X chromosome and a Y chromosome, and normally the Y chromosome will have a functional SRY gene. And if that's the case, then the indifferent gonad starts developing as testes, and that will then push the rest of the body to develop in a male fashion, as we'll describe in a moment. On the other hand, so questions about that? And folks asked about intersex. It sometimes happens that because remember, there's recombination takes place in gametogenesis during meiosis, where sometimes parts of two homologous chromosomes will swap bits. And that happens all the time for autosomes. It's important. That's the driver of evolution, all that recombination, so that every gamete you make has both some of your mother's genes and some of your father's genes on every chromosome, except the Y chromosome. So there's some regions of the X and Y that still swap bits, and they're not terribly important. And this is sometimes known as the pseudo-autosomal region of the chromosomes. But because some swapping does take place, it sometimes happens that an SRY gene ends up onto an X chromosome. Which parent might this happen in? It's got to be the father, right? Because he's the only one that's got an SRY gene. Sometimes in making a gamete, his SRY gene will do a crossover event and end up on the X chromosome, and that forms a gamete. And if that's the one that fertilizes the egg and a female results, and so an XX individual result. And in those cases, what we're going to see is the indifferent gonad forms as a testis, and everything else about this individual will look male. So that one gene in mammals causes a cascade of events that determines this individual is going to be male. We can expand that to say that the decision about whether an X or Y chromosome is there, we can refer to that as sex determination. That's the point at which sex gets determined. And in the normal course of events, everything else will follow. In situations like this, we can be more refined that it's SRY gene, one gene, that is important for sex determination.
Following the infertile.
Yes. So these individuals, it turns out for meiosis to take place, anything that interferes with meiosis, like odd chromosomes, means that they won't be fertile. So that's one reason. They can't make functional gametes. What's the other reason why they can't? Right? Well, we'll talk about it. We'll talk about that later. Yeah.
Compositions XX.
Sometimes it doesn't happen because most people go through life, you never get a karyotype, right? Have any of you ever had a karyotype made? Most not. But it would cause a result of sterility, right? So they might report to a doctor because they're trying to have a kid and their wife's not getting pregnant. And if the doctor, most doctors wouldn't. It would be, well, what are you getting atropine levels like? Give me a semen sample. And I guess one of the things they'll see there is there'll be very little viable sperm. Another question? Yeah. And that's probably as far as it goes. That's a great question. I don't know. I don't know if they produce any because chromosomal aberrations really interfere with gametogenesis. So I don't know. I have to look it up. I'm starting to have doubts. On the other hand, what sometimes happens is that a male will fertilize an egg with a Y-bearing sperm, but for whatever reason, the SRY gene is dysfunctional. And if that happens, the indifferent gonad won't be getting SRY. And so you'll end up with an individual that has male genetic sex, but the rest of the body looks female. And here we have female genetic sex, but the body is male. This is just the beginning. We're going to be talking about lots of situations like this. Because while there's a normal or modal pathway, what we'll see is, as in any chain of events, there are genes that are required that must work at the right point in the chain for the rest of the events to unfold. And sometimes the genes don't work, and therefore the events unfold in an atypical fashion. And in the early 20th century, we learned that SRY is itself under genetic control. So what normally happens is there's a gene that's on the X chromosome called DAX1. And everybody's X chromosome has a copy, I mean, typically. And the job of SRY is to overrule what DAX1 would normally make. And females, on the other hand, because they have two copies of DAX1, that pushes them down the pathway of making an ovary. But it sometimes happens that an X chromosome may have a duplicate DAX1. So the one X chromosome, mom provided it, and there was a stutter. So now there are 2 copies of DAX1, and therefore it's going to make twice as much or more DAX1 protein than it normally would. And in that case, even if dad provided an SRY, the DAX1 will overrule. There'll be too much DAX1, and a female gonad will form. This tells us that part of how SRY works is because it doesn't provide another copy of DAX1. But that can't be the only reason, the only thing it does, because remember, those individuals who have a dysfunctional SRY gene and only have one DAX1, and they make a female gonad. So the rule is, if there's equal contributions of DAX1 and SRY, a testis results. But even if there's SRY, if there's a double dose of DAX1, a female results. I'm going to the trouble to talk about all this complicated stuff to say that this means there's an idea out there, which I really despise, not only because it's simple-minded, but also it's a little bit dismissive, that everyone begins life as females, or that the female is the default sex. And we'll talk about later about why that's the case, but why that gets said. But the point I'm making here is, making a female body, it's not just like, oh, shrug your shoulders and everything will work, right? It takes just as much complicated genetic and morphological work as making any other thing. So there's no such thing as a default sex. In both cases, there has to be an active process going on to make either a testis or an ovary. After that, well, questions about that. So how is it that this one genetic decision can result in changes that affect so many different parts of the body? And the reason is because once the gonadal sex is determined, after that point, all sexual differentiation of the body is mediated by hormones. And so the question is, which gonad gets formed, and therefore which hormones get produced? And depending on which hormones get produced or not produced, then the body will take on a male or a female pattern. So in this class, we're just going to regard a hormone as a chemical that's secreted by one part of the body that goes into general circulation, into the blood, and eventually has an effect on another part of the body. And among the hormones we'll talk, so here's a diagram of it. We tend to say that the glands that produce a hormone, we call them endocrine glands because they release their products inside the body, endo, as opposed to exocrine glands like tear glands or Cowper's glands or prostates, which send their fluids out through a duct. So these are ductless glands or endocrine glands. So they make the hormone. They have to have this cellular machinery to release the hormone into general circulation. It travels in the blood, comes out of the blood around some target cell, and then interacts with that target cell. What makes a target cell a target cell? It is that it has some sort of receptor system to recognize and respond to the hormone. How's that? And the importance of testicular secretions for making a masculine body was actually worked out in the 18th century and then was repeated in a famous experiment in the 19th century using chickens. So, as you know, and it was pointed out to Mr. Coolidge, if you have a bunch of chickens, if we're trying to raise chickens and keep a flock, you really only need one male because he will happily mount all the hens and keep you in eggs and more chickens, etc. And people have known that for a long time. This means that if you have a flock of birds and you have more than one male, one of those males is worthless to you. And in fact, if your rooster is functioning right now, you can either kill the cockerels, the young males that come out, or you can do something with them. And in ancient times, Aristotle describes how to do this. People who kept chickens knew what to do with those excess males. It is, you castrate them. You take out their testes. They don't actually take them out. They would just burn them. And then something cool happened. The cockerel wouldn't grow up to be a rooster, wouldn't look like a rooster, wouldn't act like a rooster, and better yet, it wouldn't have tough meat like a rooster. And the Romans loved eating these chickens that had been castrated when they were young. What are they called? Yeah, I mean, nobody knows what they are anymore. They're called capons. And until the modern supermarket store, everybody knew what a capon was, because in general, they tend to be bigger and tastier and juicier than chickens, than hens. Everyone see that? And they're almost not made at all anymore. If you run a chicken farm today, those eggs that hatch, they're going to be roosters, are still worthless to you. But nobody bothers to castrate them anymore. Most chicken farms, when they look, did anyone know how to tell a male chick from a female? No, none of you do. Trust me. It turns out it's pretty subtle, right? Kind of have to learn how to do it. But all these chicken farms, as the chicks hatch, they have people who have learned to figure out whether that's a male or female. If it's a female, great. You're going to get eggs from that and more chickens. If it's a male, they're worthless to you, and they kill them immediately, typically by throwing them into a wood chipper. Yep. Yeah. It's fast. They can't hurt very long. And maybe you could turn it into dog food or fertilizer, things like that. Because otherwise, if they grow up, they become roosters. They fight with the other roosters. They disrupt everything, and you don't need them anyway. And you can't even eat them because the testosterone that made them so feisty has also made their muscles strong and stringy, and therefore the meat's tough. There are efforts underway now to test eggs early, don't wait until they hatch, to see whether they are male or female genetically. I'm not going to talk about, it's a different sex chromosome arrangement in birds than in mammals. And that might work out where instead, the egg just got laid. If there's an embryo in there, it's tiny. And if it's a male, we'll use that for eggs, right? We won't let it hatch. But that's expensive, so we'll see. Anyway, so my point is, people have known for thousands of years that a cockerel, if castrated while young, will be different from other roosters. When it grows up here, when it grows up, it won't have long feathers like roosters. It won't have big combs and wattles like other roosters. It won't have the spurs that roosters have on their legs to fight and kill other roosters and be the one that survives. And blessedly, they won't crow at 4 a.m, right? So people have known for thousands of years that castrating cockerels result in all these changes, so they act mostly like females. And sorry, I'm being harassed. And in the 18th century, an English physician named John Hunter did an experiment where he found that if you remove the testes, which would normally make a capon, and then it instead stuck it back inside the abdomen, the animal grew up looking like a rooster. And in the 19th century, a fellow named Berthold repeated the experiment and kept better notes and published. And he confirmed that as long as the testis was put back in the abdomen, and as long as there was a vascular supply getting to the testis, that then the animal would grow up like other roosters. It would be infertile, right? Why? Yeah. Because the testes aren't connected to the ductwork, right? There's no way to get sperm out, but they still have big waddles and they still crow and they still are aggressive. And their legs will still grow those thorns, claws, to fight each other. So you would think that Berthold then would have said, and of course, people had understood the effects of castration, especially early in life, on humans for hundreds of years already, also for thousands of years. You would think that he would then have said, oh, testes must make a hormone that goes everywhere in the body and affects the wattles and the comb and the feathers and the behavior. But he didn't, right? He somehow thought there must be some sort of vital humors or vital forces that are going between the testes, et cetera. It'd be another almost a century before anyone thought about hormones. But we now know that what the testes are doing is releasing a hormone, primarily testosterone, that is responsible for making all these changes. So one hormone from one source, the gonads, goes everywhere in the body and has many different effects at different parts of the body, all of which combine to make a male. They act in the feathers to make them take on colorful feathering. They act in the combs and the wattles. They act in the legs to make the claw, and they act in the brain to make the animal aggressive and interested in crowing all the time, typically at a really unfortunate hour. So the other reason you turned off most of your cockerels into capons is so you wouldn't be listening to animals crow all the time. Questions about that? So, which hormones are responsible? I need you to know that in this class, we'll talk about two different classes of hormones. Some hormones are proteins, and if it's a short stretch, you call it a peptide, which is just a string of amino acids. And those hormones are produced through the action of genes, same old genes that make any other protein. And we'll talk more about them later. There are other hormones, including testosterone. Oh, and these hormones, the protein hormones, for the most part, never get inside the cell. If they reach a target cell, it's a target cell only if the cell has a receptor that recognizes the hormone. So the hormone binds to the receptor. That binding process alters the configuration, the conformation of that complicated protein receptor, and that changes the intracellular portion of the protein to then cause some sort of chemical changes to take place. Those chemical changes can be pretty extensive. There's lots of things that can happen when the hormone binds to a target cell. And in some cases, it can even change gene expression, but typically at the end of a cascade of changes. And all that stuff is what you read about in cell physiology and physiology classes. Oh, and another thing that can happen is it can cause this cell in return to release a different hormone. So one hormone, let's say it's a GnRH, acts on a target cell to activate it, and this target cell, in response to the GnRH, makes and releases gonadotropins, like FSH or LH. The other class of hormones that we'll talk about a lot are steroid hormones. And all steroid hormones have this classic 4-carbon ring structure. I'm not going to talk a lot about what that means. This happens to be an estrogen, estradiol. So this complicated structure is not the product of a gene, rather it's a product of metabolism, typically of dietary cholesterol. So the organism eats cholesterol and it chemically transforms the backbone of that molecule into various hormones. Therefore, the ability to make these hormones resides on whether or not the individual has enzymes that can put the cholesterol backbone through its paces, take on a particular structure of, say, testosterone or estradiol. And sometimes these are closely related. What we'll see is in the ovary, almost all the estradiol released by the ovary, first, for a short time, is a molecule of testosterone. And so the ovary will then convert that testosterone into estrogen and release it into circulation. Steroid hormones work very differently than protein hormones. Steroid hormones, because they're tiny in comparison, right? They're barely amino acid in size, barely that size. And because they dissolve readily in fats, also unlike proteins. So they have an easy time going through membranes. So steroid hormones, once they're released into circulation, basically they enter every cell in the body. And most cells they enter, the steroid hormone comes in, and then it goes back out again, and nothing happens. But target cells are those that inside the cell have a protein receptor that recognizes steroid hormone. The two bind together momentarily, and in doing so, the protein changes its configuration and becomes chemically different from how it was before the steroid hormone hit it, bound it. We refer to this as the steroid receptor complex, where the two together act differently than either do by themselves. And I want to warn you, this is not to scale. The steroid hormone is tiny in comparison to the protein, but this is just so that you can see it. And typically what happens is the steroid receptor, once it's been bound by the steroid, it now has an affinity for binding to DNA, and it directly regulates gene expression. So the steroid receptor complex recognizes a specific stretch of DNA, binds to it, and then alters the expression of the gene next to it. So the steroid receptor may be in the nucleus or maybe in the cytoplasm, but in either case, the steroid receptor complex has to get to the nucleus to get access to DNA in order to alter gene expression. Hopefully, this is all a review for you. You've run into this in other classes. And we can refer to that stretch of DNA that the steroid receptor complex recognizes and binds and affects. We can refer to it as a response element. So that's what RE is, the response element. What we're going to find is that the developing testes in mammals produce two hormones that are important for sexual differentiation. The testes are going to produce a protein hormone that inhibits the Mullerian ducts and keeps them from developing. And the testes are also going to release a steroid hormone, primarily testosterone, and it's going to maintain and push the development of the Wolffian ducts. So sexual differentiation happens when the Y chromosome or not, sex chromosomes determine which pathway is going to be begun. Then the developing gonad, if it's a testis, starts secreting two hormones to push the body to make a male phenotype. And what we'll see is that in the absence of those testicular hormones, the body's going to take on a female phenotype. The fact that hormones are involved in physiological functioning means that we humans can make products that interfere with this process. Sometimes because we want to have an efficacious effect, we want to have some sort of treatment, perhaps, or sometimes we make something and it affects hormone signaling and we didn't mean for it to. So Here are the way various drugs can affect this process. We are here zeroing in on the receptor, whether it's a membrane-bound receptor responding to an extracellular protein hormone or an intracellular receptor that's responding to a steroid hormone. The logic of how drugs have an effect is basically the same. So normally, the hormone, it recognizes and binds a receptor site and changes the conformation of that receptor to have some biochemical result. Sometimes it happens that we can make a compound that is enough like the hormone that it binds the site, but is different enough from the hormone that in binding the protein receptor, it doesn't cause the right configuration change, and therefore the receptor doesn't have its biological activity. We can refer to that synthetic compound as an agonist. Pardon me, I've gone too fast. This agonist, it binds to it just like the normal hormone, but it has a super effect on the protein. So it's even more effective than the endogenous hormone at having a biological effect. You can think of it as being, an imitation that's better than the original or more effective than the original. Typically, this happens because this sort of binding of hormone to receptor, one of the things that's important is that it's temporary. They bind briefly and then they come apart again. The way to make a really effective hormone is to tinker with the shape of the drug so that it binds to the receptor but stays there longer. And in staying there longer, keeps it in that configuration longer and therefore has more of a biological effect. And so that's why we'll call that an agonist. It's acting the same way as the hormone. And later we'll talk about the opposite, compounds that bind to the receptor but keep it from being biologically active. We'll call those antagonists. And that'll be next time. If you haven't yet, might be a good idea to read chapter 4.